Okay, I want to start today by looking at some aspects of structure, and then we'll talk about muscle function, and we'll come back on Monday and be talking about how muscles actually work to cause movement, so the larger scale, macro scale movements that cause us to move in particular directions. And we've already talked a lot about movements with hydrostatic skeletons, like with tentacles. We're going to talk more about movements with rigid skeletons, either exoskeletons or endoskeletons. This is just a picture I like, a green lacewing in flight. Where I want to get us started talking about those is some of the things associated with support, protection, and movement, which is really what this section is about. I want to remind you about integument. So integument, or the outer layer of an animal. And you're already familiar with what you'd expect is a basement membrane with cells on top of that. Could be one layer, could be multiple layers. And that outer layer we refer to as epidermis. And it is an epithelial tissue layer. So epidermis, but it's an epithelial tissue layer. Basement membrane underneath, and then down here is dermis. And I've made that basement membrane much thicker than it really would be. It really would be about 1/10 of a micron, so you'd barely see it. It would be about the thickness of one of these lines, or not quite even. But it's a really important barrier. Again, we're going to come back to it again when we talk about immune function. It's a really important barrier that keeps foreign invaders out of an animal. So at places, the epidermis is broken by openings. And one of these categories of openings are cells that are secreting, for example, oils and other materials. Those cells, if you zoomed in on them, these are exocrine glands. We've been talking about endocrine glands. These exocrine glands they're having parts of themselves often blebbing off, it's called. And you get things like oils or waxes. Making a waterproof covering, making a covering again that it's hard for bacteria and viruses to get through. If you're constantly producing these oils and spreading them out underneath, bacteria and viruses tend to get trapped in them. They're less likely to make it through to the tissue underneath. So that's an exocrine gland. In us, sebaceous glands, you don't need to know the names, but we have sebaceous glands that are producing these oils. A lot of animals add an outer covering above this, and they may be doing things like producing secreting collagen to the outside, and quinone, so they wind up with quinone-tanned collagen. Optionally, they can add calcium carbonate. So they can add calcium to it to harden it more. So grow crystals with proteins in between. Again, we talked about the trade-off between the more crystalline calcium there is in there, calcium carbonate, sometimes calcium phosphate, the harder it is, but the more protein, the more crack resistance, and it adds a bit of flexibility. So there's a mix, and selection favors different combinations of either no calcium crystals at all, a lot of calcium crystals, somewhere in between. Lots of animals also have sensory hairs that again penetrate this epithelial layer. Those can be like our hairs, which are just structures made of dead cells, but they can also have their own cells within them that extend out. So some animals actually have taste receptors, or odorants, or stretch receptors that are in the hair itself, or they can be like us. And just have a sensory neuron with stretch channels wrapped around the base of it, producing action potentials anytime this hair is moved. So a major function of hairs is as sensory organs, parts of sensory organs, detecting movement from parasites crawling across your skin, or hair telling you there's a breeze nearby, or maybe something moving nearby that's causing a bit of air movement, okay? Or a very light touch. So those sensory hairs are fitting within a hair follicle. And they're actually live cells down at the base that are very high in keratin, an intracellular protein, live cells down at the base, and as new live cells come down to the base, the ones further up starve and die, and so you get this slowly growing strand of dead cells, very high in keratin, making the hair. Now, this structure, a hair follicle with something growing out of it, doesn't have to grow only one particular structure. A feather is essentially the same thing, except it's got a much more complex structure to it. So we could also have, not in the same organism, feathers in which some of the cells are branching out. So the simplest version of a feather is a central stalk with strands going out. Again, made of dead cells, very high in keratin. So that's a simple structure, but you can add even more complexity to it. You can have side branches off these side branches that lock together and form a fan, the thing we think of as a feather. Again, that's penetrating out so here's a follicle that's growing a feather. So we tend to think of hairs and feathers as very different structures. Fundamentally, they're coming from very similar structures that are just differing in how they're organizing the cells. Okay, what about a scale? Again, here's an epithelial layer, an epidermis, and again, there's a similar structure for a scale, and a scale can grow in two ways. It can penetrate from the dermis out through the epidermis and form an external protective covering. So on mammals that have scales, they're actually modified hairs, but they're doing the same thing, or modified groups of hairs, and they're going out through the epidermis and forming an outer layer. But another possibility for a scale is to never go through the epithelial layer. This is actually typical of fish scales. Okay, they actually remain underneath the epidermis. So when a scale is taken off, it actually tears the epidermis and takes that with it. And the fish has to grow a new epithelial covering wherever that scale came off. Okay, so what you're thinking about, so for animals with an integument, we've got a common set of structures. We wind up with a really diverse array of what the integument looks like, but we have a common set of structures and genes that are creating all of that. An epidermis, a dermal layer underneath. The dermal layer underneath is doing things like holding sensory organs, producing scales, feathers, hair, producing oils. Okay. It's also the second line of defense. If something does make it through that basement membrane, there are immune system cells in there. Okay, and together making up actually your largest organ, integument. And then the things that create a lot of the variety we see in animals, hard outer coverings, flexible outer coverings, feathers, fur, scales, it's the same set of underlying structures doing similar things. Okay, questions about that? Okay, I wanted to show you some images of something you're less familiar with. We've talked about it a bit before, and the details are not that critical. I do want you to have a sense of what this looks like, so you don't need to memorize all of this stuff. But chitin is sugars with crosslinks, so simple carbon rings, just a sugar, food. But with these crosslinks that make the structure very tight. It means that an enzyme cannot get around it. Okay? It would be like you trying to pick up a piece of this floor. You can't get your hands around it. If there was a single splinter sticking up, you could grab that splinter and pull it out. Okay? But as long as it's smooth like that, you can't get your fingers around any of the strands of wood in there. You can't pull them off. That's essentially what's happening with chitin. Enzymes can't get at them. The chitin is fairly stiff, and it's surrounded with protein. So there's a protein that surrounds some of these strands of chitin. For the chitin, we're going from somewhere around 10 angstroms, around a nanometer. Now we're at 3 nanometers here. So strands of chitin tightly linked together, tightly attached to protein. Protein often quinone tanned. And then those are further assembled into strands. Those strands have, for reasons I couldn't explain, except that this hexagonal structure is pretty strong, are arranged in these hexagonal structures. So what it may do is allow strength, but be lighter than something that is solid. And then those in turn are arranged on top of each other so that each new layer is at an angle to the one below. Okay. And what that does is provide strength in all directions. You can't easily split it apart in any particular direction. And then if you looked at those then are assembled into layers And here's an electron micrograph showing you some of those layers. Denser, the outermost part is denser, the lower part is not quite as dense. When arthropods molt their cuticles, they often digest the lower layer. Not so surprising that it's a little less dense. So they recover materials in the lower layer, but they don't recover the materials in the dense outermost layer. The same things that make it hard for invaders to attack and degrade the outer exoskeleton make it hard for an arthropod to recycle it. So one of the limitations we're going to talk about for arthropods is, in fact, anything that molts a cuticle, is that it's expensive. Every time you want to get to a bigger size, you've got to throw away a lot of your resources. I mean, imagine if every time you wanted to grow, you had to take out all of your bones, throw them away, and grow a new set. Okay, that's a lot of calcium, a lot of protein that you're just throwing away. So what you wind up with is a flexible composite material. The exocuticle is often quinone tanned, and it can be calcium carbonate or calcium phosphate hardened. Okay, usually calcium carbonate for an exoskeleton. Okay, I want to jump ahead and now start talking about movement. And these are some details you definitely want to know. So the piece I just talked about, I want you to have a sense of the structure of these, and I might have an exam question where I ask you to speculate about structure of a cuticle or tell me what's a logical arrangement. Here's something you found. Does it actually make sense? Would selection favor it? So I want you to understand it, but you don't need to memorize all those details. For movement, the molecular details really matter, and they're used over and over again in different contexts for animals, so I do want you to remember this. So we've already talked about movement on the scale of extending a pseudopod. This version of myosin is arranged with myosin in bundles. So we're looking at a number of myosin molecules. Here's how the individuals look like. A long strand, it's called the tail, and then the head. And so I'm going to talk about myosin and myosin heads. We won't refer to the tails much. But the tails wind up being the part that enables these myosins to bundle together. So they will attach by hydrogen bonding to each other. Okay, so we have individual myosins doing this. They're actually a dimer, two wrapped around each other, but we're not going to worry about it. We're just going to treat them as if they were a monomer, because the function, for our purposes, doesn't matter, the difference between them. And each myosin has an actin binding site and an ATP binding site. And exactly where they are doesn't matter to us. Well, we do care that the actin binding site is at the tip. But the ATP binding site, we're not too concerned about. Okay, so that's a bundle of myosins. If we looked at the inside of a skeletal muscle cell, we'd see an arrangement like this, in which we've got all these myosins here. So the reds here are myosins, and you can see all the little heads sticking out. And I want you particularly to notice that they're oriented in different directions. The ones at this end are all oriented like so, and the ones over here are oriented like so. And what the myosin heads are going to do is attach to an actin and then move, pushing, okay, pushing or pulling, depending on how you look at it, but moving that actin. So that's the basis for movement. If we looked at this particular diagram, if all these myosin heads are binding and moving like so, then these actins are pulled toward the center. Make sense? Okay, so that myosin heads are binding and moving. So So when they attach to an actin, that actin strand moves toward the center, and on the opposite side, we're also moving this to the center. The result is this structure called a sarcomere. Okay, that sarcomere will get shorter. Does that make sense? It's like you have, the actins are like this, and the myosins would be in between my fingers. And when the myosin heads attach, they are going to pull along each actin strand and pull them together. Okay? That's what makes a muscle shorten. Okay? Run through that with your neighbor just for a moment. Tell your neighbor what would happen if you had half as many myosin molecules, half as many myosins. So what would be different? So mutation in the regulatory region, you're making less myosin. What would be different? Okay, what would be different? So what would be different? Destiny? Okay, so less dramatic of a contraction or less of a contraction. So less contraction with half as many heads, you might not get as far. You could also argue it's going to be slower. So maybe you could, depending on where those heads were, if every myosin strand was just as long, it would just be a slower, weaker contraction. Okay? But if the myosin strands were not as long, you wouldn't get as far. You wouldn't contract quite as much. Okay? Both of those would be good answers. Okay? Other thoughts? Anything you want to check? Okay, what if if you made the affinity of the myosin head for actin much higher? So make it high, very high affinity. What would be different? Tell your neighbor. Okay, what would be different? Okay, so the initial stage might be a little faster, attaching. But if the affinity is much higher, it's going to take a little longer or even a lot longer. If it's very high affinity, it may never release. So that first attachment may be great, very fast. You get the first movement, and then you're stuck. I'm mimicking it right now, but not for long. Like I told you before, I never tell these jokes for you. Simeon? So would that make muscle contraction last longer? Right, however far you got, it would last longer. One of the ways that, we're going to come back to this, but there's selection on different types of muscle to be faster or slower, to use more energy or less energy. And some muscles that, for example, hold some of the muscle cells that hold the two valves of a clam together, the ones you guys looked at in lab, some of the fibers in there actually cycle really slowly. The myosin heads cycle really slowly, and they stay attached for a long time, which means you wouldn't want to use them to close the shell, okay, because they're really slow. But once some other muscle has closed the shell, you can use those to hold it tightly closed, because the myosin heads are hanging on so long that it doesn't take you very many ATPs. Okay, if the cycling rate is really low and the affinity is high until you cycle the ATPs, you can use low energy but still hold that shell closed. Okay. So the next time you eat a scallop, I want you thinking about the fact, oh, there's two different types of muscle fibers in here, and I can taste the difference. Well, okay, maybe you can't. Okay, the structure of the actin. So you looked at this in the video, but this is neater than my video. The actin are these little green things here. Okay, each of those little green things are actins. Okay, I think it's an actin dimer, each double little thing here. And they're together forming this twisted strand of actins. In the sketch, I tend to just show one because it's a lot simpler, and these pale blue spots are the binding sites for myosin in this schematic. But in skeletal muscle, those are not normally exposed. In skeletal muscle, this long, thin protein, tropomyosin, in purple here, covers, and you can see it here, covering binding sites. It's in the way physically, so myosin cannot bind. Okay, so for most of your muscles, as you're sitting here, most of your muscles are relaxed, and that's the state they're in. Tropomyosin is covering up the binding sites of actin. These little red things here are troponin, a calcium-binding protein, and actually some of the calcium-binding proteins we've talked about in other contexts are relatives. They're in the same protein family, same gene family as troponin. Okay. So here's troponin, calcium-binding protein, and when calcium binds, calcium plus 2, it's going to change the shape of troponin. When it changes the shape of troponin, troponin is attached to tropomyosin, and that change in shape pulls the tropomyosin to one side, exposing the binding sites. So that's what allows contraction to take place. So as you're moving your pencil, you are selectively releasing calcium into the muscle cells where you want contraction to take place. Okay, so in the absence of calcium, no binding. Here's ADP, and here's a phosphate. Okay? And this is in what's called the cocked position. Ready to go. When calcium comes in, so when calcium is present, that shifts the tropomyosin, the binding site is exposed, and myosin head binds, phosphate pops free. So with the new conformation, that changes the shape of the binding site for phosphate. It doesn't fit neatly anymore, and out pops the phosphate. And our myosin head has moved. So it was here, and now it's moved, and it's moved the actin. Okay, now with the phosphate gone, the ADP is no longer stably bound. And so first, phosphate pops off as we move, and then second, the ADP. Okay, so what powered the move? Well, when the phosphate was still bound, there was a -2 charge there, right? When myosin bound, that meant the binding pocket for phosphate no longer holds the phosphate, so out it pops. We've lost the -2 charge. That +2 charge in this area is what caused the sudden change. Okay, somewhere else, presumably, there's a negative charge that's pulling that over. Okay, so it's the charge that is causing the movement. Okay, once the phosphate is gone and movement has happened, now the binding pocket for ADP no longer holds ADP. So 1, the phosphate leaves, 2, the ADP leaves, but the myosin head is locked, attached to the myosin until it binds, to the actin, until it binds a new ATP. Okay, it's called the rigor position when it's in this position. Rigor comes from rigid and actually comes from rigor mortis. So the reason muscles get stiff after an animal dies is that as ADP, as the muscle cells start to deteriorate, calcium gets into the cells, that triggers this movement, and then over time, the mitochondria are not getting in enough oxygen, they're not able to make new ATP, so over time you run out of ATP, you eventually get to a state where all the myosins are attached to actins and they can't get free. And so the muscles become rigid. And then as enzymes break free and start to break down the cells, eventually you start getting the cells breaking down and then the muscles loosen up again. But during that initial period, they're rigid, so it's called the rigor position. So how do we get out of that? We get a new ATP, comes in and binds. With that ATP attached, that then changes the conformation of the tip of the myosin, and it can pop free. And then once it disattaches, 6, the ATP separates, the binding pocket changes such that that last phosphate is pulled off. And 7, that phosphate, the charge, moves the myosin back to the cocked position. Okay? So part of what I want you to get, the big thing I want you to get from this is it's the phosphate, it's the charge on that phosphate that is powering this. Okay? When the phosphate is there, ADP and phosphate, you get it in the cocked position, take off that phosphate, okay, change the conformation so the phosphate is no longer held there, take off that phosphate, loss of that -2 charge at that location causes it to move. How do you get it back to the cocked position? You got to get another phosphate in that position. To get to that, in order to get a new phosphate in that position, you first have to get that ADP out of the way, but myosin is still bound. You need a new ATP to bind. That changes the conformation such that the myosin head can pop free. So myosin disattaches. Now a new ATP can come in there. Sorry, new ATP comes in, myosin disattaches. Now that ATP, that change in the binding pocket for ATP splits off that PO4 -2. And now that PO4 -2 gets it back into the cocked position. Okay, what I want you to do is try and close your eyes and step your way through all of those events. I'm going to zip ahead to an image that I think has all of them together. I think I have it. Not quite. Okay, so keep closing your eyes, trying to go through it, opening them up when you need to. Okay, if it helps you to think of it without attaching the actin there, I've got a really ugly sketch. Looks even more ugly when I blow it up. Here's a myosin with an ADP and a phosphate. It's in the COC position. That phosphate is off by itself. Okay, if that phosphate pops free, triggered by binding to actin, if that phosphate pops free, it goes to the rigor position. We just have an ADP. The ADP can now pop off. The change in conformation means ADP can leave, but it stays stuck till we get a new ATP, stays in the rigor position until we split the ADP and phosphate, and now we're back. Okay? So that's the cycle of events powered by that PO4 minus 2 being put on, taken off, put on, taken off. Okay? It's as if we've flipped a switch, okay? Put on the minus 2, and we're attracted out here. Take it off, we're back here. Put it on, we're back here. Take it off, we're here. Okay, any questions? Zach?
So is there always enough tension on the actin itself that it doesn't slip back when the...
So what happens at the end of this is basically what you're wondering. So once you take the calcium out, okay, once the calcium is removed by calcium pumps, once the calcium is gone, that covers up, the tropomyosin covers up the actin binding sites for myosin, and the process is going to stop. And so the question is, will it just go back, or will it stay shortened? And if you think about it, it took us energy to get it to shorten. The only way it can go back is if there's some kind of elastic recoil. Usually there isn't, or there's very little elastic recoil. So contract a muscle, it'll just stay contracted until an opposing muscle, okay? So if I contract my biceps, okay, and I have it like this, it just stays contracted until an opposing muscle, my triceps, pulls it out, okay? You do have some muscles that use elastic recoil. When we're running, we actually get some elastic recoil in our Achilles tendon. Kangaroos get a lot of it. But for the most part, once you contract the muscle, it just stays shortened until an opposing muscle stretches it out again. Okay, make sense? Yeah. Angie? Yeah. Yeah, so are the heads doing, so in these bundles, are the heads doing this motion all at the same time? So if one of these bundles was here, some would be here, some would be oriented this way, and this way, and this way, all the way around, with actins all around them. And they're all going through this cycle at the same time. What limits it is they can only move once they attach to an actin. So there has to be an actin available for them to touch. But if there is one, they'll cycle through, pop free, and go back, as long as there's ATP. So all these myosin heads, we'd be doing it at once. So if you could see them all going, it would be this crazy movement of all these different myosin heads at different locations, doing different things, okay? But all helping to pull the two ends of the sarcomere together, to pull the actins together. Meg? So if every individual myosin head requires its own ATP, how is this not too wildly expensive? It is wildly expensive. You need a lot of mitochondria. In hardworking muscle, you need a lot of mitochondria in there to power them with the ATP. Now, how expensive is it relative to other things? Your brain is using as much ATP as hardworking muscle. So all of the action potentials you're producing, all of those events, powered by the sodium-potassium pump creating the concentration gradients, that's also really expensive. So in comparison, it is expensive tissue when you're working hard, but so are some other things. And this is why you need a lot of mitochondria to power it. Okay, Sarah, did you have a question?
Yeah, just a clarification. So the phosphate leaving, it causes a change in conformation in the myosin, and that's what causes ATP.
So the question is, a clarification, the phosphate leaving is what causes the movement from the COC to the rigor position, and that's correct. You take off that -2, you can predict that's going to change its shape, right? And then that change in shape is somehow constricting the binding site for ADP, right? So when it makes that movement, it's constricting the binding site for ADP, so ADP pops off, okay? Okay, Simeon?
So when you talk about calcium enters
right? Channels allow it in. So we'll come back to that. So so we're going to modify what I said before. It's the same story, but calcium pumps pump calcium out of the cytoplasm, okay, out of the cytoplasm. That can be out of the cell, or it can be into an internal calcium storage location. So you guys all remember the nuclear membrane is a double membrane. Okay? In many cells, the space between the two nuclear membranes is a calcium storage location. And so calcium pumps are moving calcium into it, and channels will allow it out as a second messenger. Sarcoplasmic reticulum is a modified version of endoplasmic reticulum, and it is modified as a calcium storage location. Okay? So calcium pumps, and good point, this is a good point to bring it up. It's going to come back later, and it's always a point of confusion. Calcium pumps move calcium out of the cytoplasm. I don't know any exceptions. Calcium pumps always move calcium out of the cytoplasm. That can be out of the cell completely, or it can be out of the cell but into a calcium storage location. Okay. And sarcoplasmic reticulum, it sort of trips off the tongue neatly, but I remember it being painful to learn. Sarcoplasmic reticulum is one of those calcium storage locations. Okay, good question. Okay, I think I saw another hand somewhere in that region. Maybe not. Oh, yeah, Tanner.
Why are the contractions, why does it start quickly if it pumps in and there's channels out?
Okay, so why does it start quickly? It pumps out of the cytoplasm, channels into the cytoplasm, okay? So channels to let it into the cytoplasm. If you open a channel, all you have to do is wait for a calcium that's bouncing around out here to hit it and go through. So that's opening a channel is fast. A pump, getting it back out of the cytoplasm. A pump, you got to wait for calcium to bind. You got to wait for an ATP. You got to wait for the ATP to be cleaved. You got to wait for the protein to change shape. Pumps are slow.
Okay, I thought the video said that there was a calcium pump that pumped it into the cell.
The calcium pumps pump it into the sarcoplasmic reticulum. That's a calcium store. So the pumps are always taking it away from the myosin and actin. Channels are letting it in. Pumps are moving it. So the start of muscle contraction is fast because that's based on channels. Okay. Ending muscle contraction is slower because that waits for pumps. Okay? Wait. Charles. Okay, yeah.
How does troponin take away tropomyosin so that myosin is fine?
Okay, so how does troponin take away tropomyosin? So if I'm a troponin and I'm attached to this strand of tropomyosin here, okay? So I've got a calcium binding site and then I'm attached here. If we put a Ca+2 on me, first of all, if I've got a binding site for calcium, it means I'm negatively charged, right? And if suddenly we neutralize that negative charge with a calcium +2, that's going to change the shape of the rest of the protein. And so I pop free, and that allows the tropomyosin to move out of the way. Okay. Now, the other possibility is that maybe when the calcium binds, instead, I push the other way and I shove all these people out of their chairs, okay, freeing up the binding sites for myosin. Okay. Either way, it should make great sense to you that a calcium-binding protein is going to change shape if we put a calcium on it, and that can force something else to move. Okay? Okay, good questions. Okay, so here is a pair of sarcomeres. By the way, sarc or sarco is either a Latin or Greek root for muscle. This is where it comes from. Hence sarcomere, sarcoplasmic reticulum. So here are two sarcomeres, and notice that here there's not much overlap of the actin and myosin. The actins end right here and here. And if we let calcium in, contraction takes place. We've moved these actin strands, and now, in fact, there's complete overlap. Okay? As you might guess, something we won't talk a lot about here, but as you might guess, when contraction starts with only a few myosin heads overlapping, right? These can't do anything. Gets back to the question earlier, I think, from Jessica. These can't do anything. These guys can move, so you're actually going to get a weaker contraction at this point. There's fewer myosin heads involved. Whereas by the time you get close to here, you've got a stronger contraction because every single myosin head can be active. You actually have different strength of contraction when your muscles are completely stretched out versus when they're partly shortened. Okay, so that's how muscles shorten. So if you have a whole bunch of sarcomeres in a row, and they're all shortening, the muscle cell will shorten. Okay, that's what the muscles do. All right, let's put these together into a muscle and then start talking about how we control this. So put into a muscle, here is a sarcomere. These reds represent the myosin. Here are the actins, the dark gray. Here's one sarcomere, and they're in these cylindrical bundles. So if you looked at the edge or you cut through it, you'd be cutting through myosins, and every bundle of myosin is surrounded by actins, and every actin has myosins around it. Those, here we are, here's one of those tubes, here's another one, so there's 5 of them shown here. Okay, here's one where you can see more of it. And in order to make muscle contraction happen fast, you need the calcium to be really nearby. If you have to wait for calcium to get from the outside of the cell, the muscle contraction is going to be a lot slower. So here's the sarcoplasmic reticulum, all this reddish stuff. Okay? That is storing the calcium. So the calcium is close to every single sarcomere. You're going to get calcium in there quickly once you open those channels. This represents the membrane around the cell. So this pale part here. And then these are the T-tubules, the transverse tubules coming in here. So the membrane actually continues. This is part of the membrane. And if an action potential spreads across this membrane, it'll go down these little tubules. Okay? So an action potential goes over, not just over the outside of the cell, but you get a voltage change down by these tubules, and that opens voltage-gated calcium channels both in the membrane itself and all along the sarcoplasmic reticulum. Okay? So there are voltage-gated So with an action potential, calcium comes out and floods the area around every single sarcomere. Fast contraction. Now, once the action potential ends, calcium pumps, which are constantly running, calcium pumps are going to be moving that calcium back into the sarcoplasmic reticulum. Okay, so calcium coming out from an action potential going back in. These are some of your biggest cells. You can have individual cells, like in your thigh muscles, can be centimeters long or even 10 centimeters long. And they are multinucleate. They actually start off as smaller cells and fuse. That detail doesn't matter so much to us. The real point is a single action potential will contract the whole thing, the whole cell. And it does it by opening voltage-gated calcium channels along these T-tubules. Okay, to finish off today, these are assembled. So here's one multinucleate muscle cell. Now in small animals, they're not multinucleate. In small animals, it's a single nucleus. They're much smaller cells. In your intestine, smooth muscle, much smaller, still long and slender, but smaller cells, single nucleus. But these are big. So here's a muscle cell with a couple of nuclei seen here. These are the individual sarcomeres inside. And then here's a bundle of muscle cells. And when we say muscle fiber, that means muscle cell. It looks like a thread, so they get called muscle fibers. All assembled into a muscle. All this orangey stuff, yellow stuff around here, that's all connective tissue. That all holds it together so that it's reasonably tough. Okay, we'll come back next time to talk about control, and then we'll talk about muscles controlling lever systems to cause movement in animals like us.